5.3 Active transport. By the end of this section, you will be able to do the following. Understand how electrochemical gradients affect ions. Distinguish between primary active transport and secondary active transport. Active transport mechanisms require the cell's energy, usually in the form of adenosine triphosphate, ATP. If a substance must move into the cell against its concentration gradient, that is, if the substance's concentration inside the cell is greater than its concentration in the extracellular fluid, and vice versa, the cell must use energy to move the substance. Some active transport mechanisms move small molecular weight materials, such as ions, through the membrane. Other mechanisms transport much larger molecules. Electrochemical gradient. We have discussed simple concentration gradients, a substance's differential concentrations across a space or a membrane, but in living systems, gradients are more complex. Because ions move into and out of cells and because cells contain proteins that do not move across the membrane and are mostly negatively charged, there is also an electrical gradient, a difference of charge, across the plasma membrane. The interior of living cells is electrically negative with respect to the extracellular fluid in which they are bathed, and at the same time, cells have higher concentrations of potassium, K+, and lower concentrations of sodium, Na+, than the extracellular fluid. Thus in a living cell, the concentration gradient of Na+ tends to drive it into the cell, and its electrical gradient, a positive ion, also drives it inward to the negatively charged interior. However, the situation is more complex for other elements such as potassium. The electrical gradient of K+, a positive ion, also drives it into the cell, but the concentration gradient of K+ drives K+ out of the cell, figure 5.16. We call the combined concentration gradient an electrical charge that affects an ion its electrochemical gradient. 5.3. Active transport. 143 Visual connection. Figure 5.16 Electrochemical gradients arise from the combined effects of concentration gradients and electrical gradients. Structures labeled A represent proteins. Credit. Synaptitude. Wikimedia Commons. Injecting a potassium solution into a person's blood is lethal. This is how capital punishment and euthanasia subjects die. Why do you think a potassium solution injection is lethal? Moving against a gradient. To move substances against a concentration or electrochemical gradient, the cell must use energy. This energy comes from ATP generated through the cell's metabolism. Active transport mechanisms, or pumps, work against electrochemical gradients. Small substances constantly pass through plasma membranes. Active transport maintains concentrations of ions and other substances that living cells require in the face of these passive movements. A cell may spend much of its metabolic energy supply maintaining these processes. A red blood cell uses most of its metabolic energy to maintain the imbalance between exterior and interior sodium and potassium levels that the cell requires. Because active transport mechanisms depend on a cell's metabolism for energy, they are sensitive to many metabolic poisons that interfere with the ATP supply. Two mechanisms exist for transporting small molecular weight material and small molecules. Primary active transport moves ions across a membrane and creates a difference in charge across that membrane, which is directly dependent on ATP. Secondary active transport does not directly require ATP. Instead, it is the movement of material due to the electrochemical gradient established by primary active transport. Carrier proteins for active transport. An important membrane adaptation for active transport is the presence of specific carrier proteins or pumps to facilitate movement. There are three protein types or transporters, figure 5.17. A uniporter carries one specific ion or molecule. A symporter carries two different ions or molecules, both in the same direction. An antiporter also carries two different ions or molecules, but in different directions. All of these transporters can also transport small, uncharged organic molecules like glucose. These three types of carrier proteins are also in facilitated diffusion, but they do not require ATP to work in that 144 Chapter 5, Structure and Function of Plasma Membranes. Access for free at openstax.org. Process. Some examples of pumps for active transport are Na plus K plus ATPase, which carries sodium and potassium ions, and H plus K plus ATPase, which carries hydrogen and potassium ions. Both of these are antiporter carrier proteins. Two other carrier proteins are Ca2 plus ATPase and H plus ATPase, which carry only calcium and only hydrogen ions, respectively. Both are pumps. Figure 5.17. A uniporter carries one molecule or ion. A symporter carries two different molecules or ions, both in the same direction. An antiporter also carries two different molecules or ions, but in different directions. Credit. Modification of work by Lepasque, Wikimedia Commons. Primary active transport. The primary active transport that functions with the active transport of sodium and potassium allows secondary active transport to occur. The second transport method is still active because it depends on using energy, as does primary transport. Figure 5.18. Figure 5.18. Primary active transport moves ions across a membrane, creating an electrochemical gradient, electrogenic transport. Credit. Modification of work by Mariana Ruiz Villarreal. One of the most important pumps in animal cells is the sodium-potassium pump, Na plus K plus ATPase, which maintains the electrochemical gradient, and the correct concentrations of Na plus and K plus, in living cells. The sodium-potassium pump moves K plus into the cell while moving Na plus out at the same time, at a ratio of 3 Na plus for every 2 K plus ions moved in. The Na plus K plus ATPase exists in two forms, depending on its orientation to the cell's interior or exterior and its affinity for either sodium or potassium ions. The process consists of the following 6 steps. 1. With the enzyme oriented towards the cell's interior, the carrier has a high affinity for sodium ions. 3 ions bind to the protein. 2. The protein carrier hydrolyzes ATP and a low-energy phosphate group attaches to it. 3. As a result, the carrier changes shape and reorients itself towards the membrane's exterior. The protein's affinity for sodium decreases and the 3 sodium ions leave the carrier. 4. The shape change increases the carrier's affinity for potassium ions, and 2 such ions attach to the protein. Subsequently, the low-energy phosphate group detaches from the carrier. 5. With the phosphate group removed and potassium ions attached, the carrier protein repositions itself towards the cell's 5.3, active transport 145 interior. 6. The carrier protein, in its new configuration, has a decreased affinity for potassium, and the 2 ions moves into the cytoplasm. The protein now has a higher affinity for sodium ions, and the process starts again. Several things have happened as a result of this process. At this point, there are more sodium ions outside the cell than inside and more potassium ions inside than out. For every 3 sodium ions that move out, 2 potassium ions move in. This results in the interior being slightly more negative relative to the exterior. This difference in charge is important in creating the conditions necessary for the secondary process. The sodium-potassium pump is, therefore, an electrogenic pump, a pump that creates a charge imbalance, creating an electrical imbalance across the membrane and contributing to the membrane potential. Link to learning. Watch this video, https://openstax.org/l/na_k_atpase to see an active transport simulation in a sodium-potassium ATPase. Secondary active transport, co-transport. Secondary active transport brings sodium ions, and possibly other compounds, into the cell. As sodium ion concentrations build outside of the plasma membrane because of the primary active transport process, this creates an electrochemical gradient. If a channel protein exists and is open, the sodium ions will pull through the membrane. This movement transports other substances that can attach themselves to the transport protein through the membrane. Figure 5.19. Many amino acids, as well as glucose, enter a cell this way. This secondary process also stores high-energy hydrogen ions in the mitochondria of plant and animal cells in order to produce ATP. The potential energy that accumulates in the stored hydrogen ions translates into kinetic energy as the ions surge through the channel protein ATP synthase, and that energy then converts ADP into ATP. Visual connection. Figure 5.19. An electrochemical gradient, which primary active transport creates, can move other substances against their concentration gradients, a process scientists call co-transport or secondary active transport. Credit. Modification of work by Mariana Ruiz Villarreal. If the pH outside the cell decreases, would you expect the amount of amino acids transported into the cell to increase or decrease?